This is Jose Merino, Editor-in-Chief of the Neurology family of journals. The Neurology podcast provides practical information to neurologists and other clinicians to help them provide better care for their patients. Thanks for listening and have a great week.
Hi, everyone, and welcome to the Neurology podcast. My name is Deanna Saylor, and I am an Associate Professor of Neurology at the University of North Carolina, Chapel Hill, and editor of Without Borders, the global health section of the Green Journal. Today, we are talking with Senjuti Saha about her recent Viewpoint article published in Lancet Digital Health titled Overcoming Colonialism and Pathogen Genomics. Dr. Saha is a Deputy Executive Director of the Child Health Research Foundation in Dhaka, Bangladesh. Welcome to the Neurology podcast.
Deanna, thank you so much for inviting me.
Your recent Viewpoint really covered two topics, which not all may be familiar with: colonialism and global health, and also pathogen genomics. So before we begin discussing your article in detail, I wonder if you could just tell what is meant by the term colonialism when it applies to global health.
I'm going to start with the broad definition of colonialism, and then I'm going to get into health. So colonialism really means when one powerful group or a country takes control over another group or a country. Usually it's land, resources, and often it's people. And usually the land, resources, and people are taken over for economic or political gain. In health and science, or overall in global health, colonialism means decisions, money, and knowledge stay with powerful countries or powerful institutions, while really the places most affected by diseases often have little to say, have little funding, and have very little access to tools or training. Now, colonialism and global health is actually quite an old concept. We heard about tropical medicine centuries ago. This was really when countries from the West colonized countries in the global South, or the global North colonized the global South and tried to protect themselves from the diseases of the global South, so that they could enhance their colonization of those lands. At that time, scientists, doctors were trying to protect themselves from malaria, from yellow fever, and global health at that time was called tropical medicine. Gradually, that evolved to become international medicine, but really to protect against diseases of the global South. The term now used often is global health, but encapsulates the same concept. While global health in theory should mean health for all the people of the world, if the United States and Canada are working together on a health project, it's not called global health. But if US is working on, let's say, a project in Malawi, it is called global health. So that is perhaps an explanation of colonialism and global health.
I do a lot of my work in Zambia, and I think it's interesting that even when my Zambian colleagues are doing work in Zambia, it is often referred to as global health, even though it's obviously very local health for them.
Absolutely. That's actually public health, like public health in any other country, whether rich or not so rich.
Exactly. And I think this is just such an important topic, and one that I certainly try to consider in all aspects of the work that I do. And now the other main topic of your Viewpoint is pathogen genomics. And as neurologists, a lot of us don't think about pathogens all the time. And so I'm hoping you can just give us a brief overview of what this is, and in particular, how it is relevant to us as a community of neurologists.
So pathogens is mostly microbes, for example, bacteria, viruses, or let's say fungi that cause diseases to animals. And all microbes have some form of code to their life, genetic code, either it's DNA or RNA. Through the latest technologies of genome sequencing, we can actually read their genome sequences and predict their characteristics. For example, if a bacteria like Streptococcus pneumoniae becomes resistant to certain antibiotics, by looking at its genome sequence, we can actually tell why or which gene or what mutation in their genomic sequence is actually making that specific strain of Streptococcus pneumoniae antibiotic resistant. So that's pathogen genomics, reading and understanding the genomes of pathogens. It's very important to understand the genomes of pathogens because by looking at their genes, reading their genes, we can understand or gain insights on transmission of different pathogens. For example, viruses. How are viruses transmitted? How do viruses target different cells? How do different pathogens actually invade our neurons? Pathogen genomics is also very important for vaccine design. In the field of meningitis, we often talk about the hib vaccine, the vaccine that protects us and protects children against Haemophilus influenzae type B meningitis, or the pneumococcal vaccine that protects against meningitis due to pneumococcus or Streptococcus pneumoniae. In order to design those vaccines, it was very important to know what do those bacteria look like. For example, what do their capsules look like so that we can design vaccines against specific capsular types. It's also important to do pathogen genomics for new drug development to target certain parts of the gene. So really with the advances and advancement of pathogen genomics, we are learning more about pathogens and how to help prevent or treat diseases.
Now that we have that background, I'd like to turn to a pathogen that we're all familiar with, which was COVID-19. And in your recent article, you really point to the COVID-19 pandemic as a turning point for pathogen genomic infrastructure and deployment of this infrastructure in several different South Asian countries. Can you highlight some of the examples that you referred to?
As I was talking about colonialism, and in health, colonized are not only lands, pieces of land and people, but also realms of health. So for a long time, this entire field of pathogen genomics had really been very unidirectional, where samples would be shipped from the global south to the global north, where the samples would be sequenced, genomes would be read, and decisions would be made, or drugs or vaccines would be designed. And in that process, we or many groups in the global south really lost out on that ability to strengthen and build capacity for pathogen genomics. But at times like COVID, when there are not planes flying from one country to another, where you need rapid data, those systems really do not work. And pathogen genomics, as we all know, was instrumental in understanding the disease COVID, the virus that causes it, SARS-CoV-2, as a whole, right? Like soon after the first cases were detected in China, because China had access to pathogen genomics, they were immediately able to figure out exactly what virus, in this case SARS-CoV-2, caused the respiratory outbreak. And so very quickly, there was understanding that pathogen genomics is very needed for dealing with outbreak, because we had pathogen genomics, because the genome of SARS-CoV-2, circulating strains of SARS-CoV-2 could be sequenced so quickly, primers could be designed, and immediately several groups across the country started designing and then developing vaccines. Although I must say that this realization came even before the COVID-19 pandemic. So during COVID-19, some of these groups who already had some infrastructure could quickly start sequencing the circulating strains of SARS-CoV-2 locally in real time. Oftentimes, these labs would be shipping their samples somewhere else in the world, but realizing that this is a need we have now and must be dealt with now, all of us came together and started sequencing as many strains of viruses as we could. So there are all of these examples where we broke the stereotype. Many countries in the global south showed that we can also sequence. It's not rocket science. When there is need and there is some support, we are able to quickly build up capacity and use it for the people.
Certainly, We have exciting metagenomics, next generation sequencing being done in the neurology field to try to better clarify etiologies of meningitis and encephalitis. And we're all waiting and worrying about the next pandemic and the next emerging infectious process that certainly could be neuroinvasive and affect us as neurologists. And so I think it's important for us to think about this as neurologists too. And despite all this progress that you talked about, you also mentioned several challenges in your article. So you mentioned limited access to the expensive machines that are needed for this work to be done. You also mentioned how the reagents are often expensive and ironically cost more in low and middle income countries than they cost in high income countries. You talked about these hub and spoke models where the hub is in high income countries like the US or Europe. The spokes are in lower and middle income countries. And yet the hubs are the ones really controlling the research agendas while the spokes are often the ones doing the work. And you talked about an internalized colonial mindset among some scientists in low and middle income countries. Can you talk more about these challenges and how we can overcome them to continue to improve pathogenomic infrastructure?
I wanted to first pick up on the point that you were making about using metagenomics for solving some of these mysterious or idiopathic cases of meningitis. Now, I was telling you that we laid the foundation for genomics in Bangladesh around 2017. And the reason we laid the foundation for pathogen genomics was exactly what you were talking about. Understanding what causes most of the meningitis cases in Bangladesh in the post-Hib and pneumococcal vaccine era. Because even today, we see a lot of children come into the hospital with meningitis, elevated white blood cell count, but in almost 80% of the cases, we have no idea what is causing meningitis. So it was to solve this mysterious problem that we first started pathogen genomics, that we started or built the infrastructure for metagenomics. And one of our first discoveries when we started doing metagenomics in Bangladesh is that chikungunya can be neuroinvasive, which we did not know, or there was no direct evidence of that before. So it was actually through metagenomics of cerebrospinal fluid samples that we realized that chikungunya virus from the blood was going into the brain and causing severe infections. Now, coming to the challenges, as we started building out this center, we realized how difficult it is. And it's also personal for me a little bit. I grew up in Bangladesh, but very early on, I actually left Bangladesh to come to North America. So I did my undergraduate training, my doctoral training, everything in Toronto, Canada. And life was pretty awesome where I could just order primers and they would show up in 24 hours. On my bench in the lab, I could just send out for Sanger sequencing, wake up in the morning and open my laptop and be able to read my results. When I moved back to Bangladesh after spending 11 years in Canada, I realized the gap. I think my memory was a little rusted and I did not realize how big the challenges were. For example, when we were trying to get our first sequencing machine, we could not directly buy from Illumina, one of the largest distributors of sequencers, because they have no local office in Bangladesh. So we had to work with a distributor who imports from Illumina into the country and then sells the product to us, which makes everything very time consuming because we order to the local distributor, they go up to Illumina, they negotiate with Illumina, a machine comes into the country, custom clearance takes forever and ever, and then we get the machine from the local distributor. Now, because there isn't a very high demand. The market for such equipment or even the regions that are needed for sequencing and other experiments are so small that it often takes a long time to get the shipment into the country. So there is really this time lag, almost, we often in our group call it wasted time and wasted motivation, because when scientists think of ideas and they're not able to do them immediately, soon either scientists get bored, move on to a new problem after waiting for months, or we just get scooped because we weren't able to act on our ideas immediately. Now, paradoxically, it's not only time that takes a toll, but we also pay a lot more. And there's absolutely no price control, which makes buying equipment and reagents very expensive. Other than that, I think one of the other questions you also had is about the hub and spoke model.
Yes. Can you tell us more about that?
As we are gradually trying to figure out how to build and strengthen capacity for pathogen genomics in our country, we're really proving to institutes in position of power, countries in positions of power, that it is possible to do genome sequencing in countries like ours. I remember when PCR first came, there was this notion that countries like ours will never be able to do PCR, design primers and do PCR because it's so complicated. But pretty much all countries now across the world have showed that's not the case. And we are breaking that barrier and mindset again when it comes to pathogen genomics. Now that we have been able to do that, what we are seeing are these initiatives where there are groups that are situated or located in high resource settings in high income countries that are trying to build these training models to train groups like ours to do genome sequencing. And there is often this idea that we are not able to train ourselves. We definitely need someone else to come and train us. Now, knowledge exchange is extremely important. We do not have the kind of training, access to the same technologies and curriculum as many of the resource-rich countries. So knowledge exchange is very important. But as I said, it's the exchange. It's not one direction of someone just coming and training us. I think it has to be both ways. And unfortunately, what we are seeing happen is this creation of hub and spoke model where, again, it is being thought that if there were hubs in some of the richer and more well-resourced countries, some of the other countries with low resources will act as spokes, where the hub will decide what happens in spokes. And we don't think that is the way to go forward, because the spokes can only be ever as good as the hub, and it really slows down all the process because all the main decisions are often made by the hub, and by the time they travel down to the spoke, decisions are no longer relevant and very slow, and there's a lot of bureaucracy and red tape. Now, the opposite of the hub and spoke model is called a point-to-point model, where all groups, whether within country or in between different countries, have their own independence of doing whatever they want in case, however they want to sequence, whatever they want to sequence. But there are risks with that too, because then there is no quality control. There is no regulation. So what we propose is this empowered network model. So we propose this empowered network model where there will be multiple groups doing work. They will be able to set their own research agenda based on the local problems, based on local expertise. However, there will be collaboration between these groups, and there will be some kind of monitoring system, some kind of a quality assurance system, who we are able to contact, who are able to contact each of these groups and ensure that work is being done.
Thanks for those explanations and really opening our eyes to many of the challenges that you face in lower resource settings, but also the incredible progress and ways you're overcoming them. And you also have a call to action for all of us. Can you talk more about what you think needs to be done going forward?
I think, first of all, our mindset needs to change, especially of scientists and government in low resource settings. We say that colonizers have left our countries, but our minds continue to be colonized. Oftentimes what I see in our countries, to this date, we believe that perhaps people located in the global north are more intelligent than we are, smarter than we are, and a training from the global north is no matter what always going to be better than, let's say, receiving training from across the street. I think it's very important that we get out of that mindset. We get out of that mindset of always wanting to ship samples out because sequencing elsewhere is better than sequencing locally. So it's very important that we get out of that mindset. We collaborate. There are such collaborations. There are in-country collaborations to really work for each of our countries, each of our communities. I strongly believe that we should be working in the community, with the community, and for the community. And in all of this, of course, we will work with our resource-rich collaborators because there is just so much to learn. But as I said before, this needs to be in the form of exchange where scientists like us who are working in low resource settings and have lived experiences have things to share about the context, the knowledge, and scientists from the global north, from more resource rich background, have the training and the expertise that they have developed, and we can exchange really our knowledge. It's also very important for governments of low and middle income countries to invest more in research. We shouldn't be constantly dependent on governments or philanthropists of other countries. And I think right now, this is a very relevant time to talk about this. There have been many recent changes. For example, the funding cuts by the NIH, the funding cuts in overall global research agenda. So it's really important that governments of countries start investing in health and research also locally, because we should not and cannot be dependent on other countries and other governments forever. I think it's also important for donors to realize the gaps in the way studies are funded. What often happens is low middle income countries in the global south do not get direct funding. There is some other institution located in a high resource setting who get the funding, and local institutes in the global south are given a sub-award. This leads to power imbalance of who decides what work must be done, who owns the data, who writes the papers, who gets the authorship. And finally, the institutions, the way scientists are incentivized or researchers are incentivized is through publication. Oftentimes, scientists who are really working with countries in the global south and helping exchange knowledge, helping build and strengthen capacity are not incentivized. So tenure track doesn't depend on how many labs have you built or how many scientists have you trained in the global south or with your collaborators. It's oftentimes based on how much grants have you brought in, or how many papers have you published, in how many of them are you the first or the last author. So I think for institutions, it's also important to realize the impact they are having. We have to move away from publish or perish idea in these global institutions or high resource rich institutions, and also incentivize scientists who genuinely are working with local collaborators and working together to build and strengthen capacity.
Thank you so much, Senjuti. You really have raised a lot of important points and given us a lot to think about in this insightful conversation. I know I've learned a lot. So as we come to a close, do you have any final words?
I imagine a world where every community, every country can quickly study the pathogens affecting that community's people. And I imagine a world where this work will be done in the community, by the community, and with the community. I imagine a world where we will quickly be able to share the data openly, but we're also able to keep the ownership and leadership locally. I imagine a world where partnerships are equal so that science is truly for everyone and everywhere.
I love this, and I want to live in that world as well. So thank you so much, Senjuti. And once again, I'm Deanna Saylor, and I've been joined by Senjuti Saha for this episode of Neurology Podcast.
This is Stacey Clardy, your podcast editor. If you've enjoyed the podcast, please take a few moments to subscribe, rate, and review the Neurology Podcast through Apple Podcasts, Google Podcasts, Spotify, or wherever you listen. And remember, you can always head to neurology.org/podcast for our full list of past episodes. Or you can also search by keyword in your podcast app for any neurology-specific topics you want to learn about.